I'm Daniel Levine, and this is the Bio Report. AI's promise in drug discovery is being held back by a simple but stubborn problem. The field doesn't have enough of the right kind of lab-generated data to train good models. Public databases and scattered studies give a patchy view of how proteins interact, mostly showing successful interactions and using different methods and conditions, which makes it hard for AI systems to learn broad rules or understand what doesn't. AlphaBio tackles this data gap with AlphaSeq, a lab platform that can test around 1 million protein pairs in one experiment under the same conditions, producing rich, consistent information on both hits and misses that's well suited for AI. David Younger, co-founder and CEO of AlphaBio, talks about why far more lab data and infrastructure will be needed than most people expect, why current public data sets fall short, and why the company's business model is built around providing data and services rather than developing its own drugs. Dave, thanks for joining us.
Danny, thanks so much for having me. Excited to be here.
We're going to talk about AlphaBio, its platform technology that combines synthetic biology and machine learning to engineer protein-protein interactions, and how its proprietary data sets are used to accelerate therapeutic development. For listeners who may not be familiar with Alpha, can you explain your platform and what makes your approach to measuring protein-protein interactions unique?
Yeah, absolutely. We spun out from David Baker's lab in 2018, and really our core innovation that led to us spinning out is a proprietary wet lab technology that's called AlphaSeq. And this is a wet lab approach for measuring very large numbers of protein interactions with quantitative resolution. So essentially, we can take 1,000 antibodies and 1,000 antigens, and in one simple experiment, we can measure all of the million pairwise affinities between those two sets of proteins. It turns out that these types of data are both really valuable for characterizing potential therapeutic antibodies and kind of advancing therapeutic programs, but also it's the exact multi-dimensional quantitative data that is particularly valuable for training highly predictive machine learning models.
We first spoke to you on the Bio Report in 2018, shortly after you spun AlphaBio out of Baker's lab at the University of Washington. How would you describe the company then versus today?
Yeah, so in 2018, really, we were two people. So my co-founder, Randolph Lopez, and myself, we were very much focused on the platform thesis and trying to ask the question, right, does this technology really work and move the needle for solving problems across pharma? At that time, we were still somewhat up in the air about whether we were going to apply the platform internally ourselves to discover our own therapeutics, or focus more on being an industry enabler and use the collective data that we generate and models, et cetera, to serve a broader set of customers and partners across the industry. So today, we are a team of 35. We've raised almost $50 million in venture capital. We have partnered with many of the leading pharma and biotechs across the industry, and we are fully committed to being industry enablers. So our role in the ecosystem is to use our data, our models, our capabilities, our know-how to advance the industry broadly. And that's both working with pharma companies and biotechs to advance individual therapeutic programs, and also to work with pharmas and model makers to essentially alleviate the data bottleneck and help them train more predictive models that are going to accelerate the industry.
Many AI protein companies emphasize modeling. A Alpha leans heavily on experimental data. Why is that?
Models are incredibly important. Don't get me wrong. We are certainly not anti-model. We build them, we use them, but our approach at Alpha is very much to be data first. We believe that there is an overall trend towards model commoditization, especially when it comes to models that are trained on public data or open source data. Uh, Really, what we believe is the differentiator and what maybe hasn't gotten enough attention is the underlying data, the quality of those data, the abundance of those data, uh, generating the right type of unbiased data that's really going to be most useful for solving particular problems. What we have seen kind of broadly as an industry trend is that companies will start by focusing on open source data, publicly available data, data that they have already collected historically, but not necessarily for the specific purpose of model training, and try to get as much as they possibly can out of those data, and then quickly hit a bottleneck. And many companies will then try to innovate on the computational side. So try different architectures and kind of different approaches for eking out as much value as they possibly can. But at the end of the day, a model is only going to be as good as the underlying data. And so we believe that that is one of the biggest challenges, one of the biggest gaps across the industry. And we're very fortunate to have a platform, a capability to be able to meaningfully address that problem for the industry.
Where do current AI models for protein engineering fall short without high quality experimental data sets?
I think one of the key places where you'll see that they fall short is generalizability. So often you'll have models that are trained on a particular data set and they're good at solving problems kind of local to that data set. So, for example, a model that's trained on the PDB is going to understand how to design protein structures and potentially binders uh, with proteins that look a lot like those proteins that they see in the PDB, but not necessarily be able to extrapolate beyond those kind of relatively small, relatively less diverse sets. So I think that that's one problem, right? Is lack of generalizability. Another major problem with publicly available data in particular is the lack of negative data. So obviously, the PDB contains the structures of true interactions between proteins. Similarly, open source data sets that do exist for binding affinity only contain positive examples where there was a measured affinity between two proteins. These databases completely lack plausible solutions that turned out not to be correct structures or that turned out not to have a measurable binding affinity. And it turns out that when you're training machine learning models, it is just as important to have an abundance of negative data as it is to have those positive examples. Another problem, kind of looking at publicly available data, the PDB is really quite unique in that the quality of data is exceptional, right? You can look at a protein structure in the PDB and expect that it's interoperable with all other protein structures within the PDB. That is just not true for a property like affinity. It depends so much on the method that was used, the buffer, the pH, the kind of precise conditions, the particular truncation of proteins that were used to measure that particular affinity. And so when you start to have these public databases that are amalgamations of data from lots of different labs, lots of different groups, lots of different approaches, the data really isn't particularly interoperable. And so the models don't do a particularly good job of understanding how to extract generalizable trends from those messy sources of data. So that's in contrast with our approach in Alpha is to use a standardized assay with standardized controls, so that all of the many billions of protein interaction measurements that we have made and train our models on, and work with partners to train models on, they are all truly interoperable.
you've partnered with major biopharma companies like Amgen, BMS, and Gilead. What specific problems are they coming to you to solve?
Yeah, so we work with pharmas and biotechs really on two classes of problems. So you mentioned Amgen, BMS, Gilead. These are companies that came to us essentially, you know, wanting to work on particular therapeutic programs, right? Solving particular problems that will unlock their ability to discover and ultimately commercialize a therapeutic. So we work with companies in the molecular glue space to discover novel targets and inform how a small molecule might enhance binding between two proteins for a molecular glue application. With Gilead, for example, they came to us wanting a more cross-reactive, more broadly neutralizing HIV therapy. So that is one type of partnership and collaboration that we have. The other type of collaboration is one where we're not focused on a particular therapeutic program, but rather we are collaborating to generate the data and train machine learning models that are really going to advance their entire pipeline. So, two very different types of collaborations, but both of them have a foundation of needing high quality, abundant data in order to solve really challenging problems for the industry.
What makes an ideal partner for Alpha today? Pharma, biotech, or even AI native model builders?
Yeah. So again, with the two different types of collaborations, one where we're focused on therapeutic programs, the other where we're focused on training better models with better data. For therapeutic programs, an ideal partner is a pharma or a biotech who has active programs where our data, our capabilities, our models will shorten cycles and allow us to engineer better therapeutics. So again, coming back to the Gilead case, they wanted a more cross-reactive HIV therapy. Obviously, that's a very important problem to solve for an indication like HIV, where you don't just want a binder to a single variant. You want a binder that's going to be cross-reactive and neutralizing against thousands of diverse variants. Um, we were able to use our data and our models to engineer a protein that demonstrated that broad cross-reactivity in a way that they would not have otherwise been able to do. That is a perfect use case, perfect partner. Um, the other type of partnership, again, is one where we're using our data to collaboratively train models and advance ML capabilities, advance those platform capabilities. And here, an ideal partner is an AI native model builder or pharma company who has really invested and bought into this thesis of uh, using AI to transform the way that they engineer protein therapeutics.
Are you seeing more interest from biopharma companies or from AI native model builders?
We're seeing a lot of interest from both. The pharma demand for data is definitely more mature. They are used to working with companies that provide services in different capacities, and so we are kind of working closely with those companies. But I would say the demand from AI native companies is growing very very fast. And as I mentioned before, they are experiencing these data walls that they're hitting into, right? They're kind of maxing out model performance from tweaks to their architecture and kind of changes to the way they do compute, and realizing that the way that they're going to get to that next level of performance is going to require access to data, and we are the ideal partner when it comes to kind of protein engineering and understanding protein binding for getting access to the type of data that's most going to change their performance.
And what have you learned about how large biopharma organizations actually adopt AI driven protein engineering tools in practice?
Yeah, I mean, I think that it's hard to generalize across all pharma. There are some pharma companies who have demonstrated a really strong commitment to the use of AI tools across a number of different functions within their organizations, and others have been slower to adopt those tools. But I think that in our experience, once we have started collaborations with a big pharma and they've gotten to see the impact of our data, uh, pretty much without fail, there's been a desire to then expand and really apply those data more broadly, more generally, with an effort of training increasingly generalizable models once they see the impact that those data can have on their therapeutic pipelines.
You've talked about the data bottlenecks in AI biology. What's missing today?
So as I mentioned, I think that there is a kind of overall scale and size limitation on what is available in the public domain. When we look at open source data like the PDB, currently there are about 1,000, fewer than 1,000 VHH bound structures with their antigens. There are about 10,000 overall antibody bound structures with their antigens. Uh, these are relatively tiny data sets compared to what kind of any other type of generalizable model would require. And so, really, I mean, thinking first just about structural data, we believe that it's going to be orders of magnitude more, on the order of tens of thousands, hundreds of thousands, even millions of examples of antibodies bound to antigens that are really going to be required to train truly generalizable models. The PDB is currently growing at a rate of about 1,000 structures between antibodies and antigens each year. And so, we do not want to wait 100 years to get to 100,000 structures. We believe that, with approaches like ours, where we can generate thousands of structures in a single experiment that takes us just a couple of weeks, we can, again, really help to relieve some of those bottlenecks and accelerate the industry.
How do you define high quality protein interaction data, and why is it so difficult to generate at scale?
So for AlphaSeq data, this is data that is highly quantitative, right? So we're not just getting binary, is this an interaction or is it not? We're actually getting a quantitative readout for the affinity of that particular interaction. We're also not just getting the positives, right? The interactions for which we can measure an affinity, but we also know the identities, the sequences of the negatives, which again is very important for training predictive machine learning models. I think what's most important when we think about quality is interoperability, right? So I am certainly not going to say that biolayer interferometry, surface plasmon resonance, quantitative ELISA, that these are, kind of bad or low quality approaches. I think when you start to run into quality issues, it's where there is a public repository of protein-protein interaction data that accumulates from all of these different sources, where different labs are using different technologies to measure affinities. They're measuring at different concentrations. They're using different truncations and variants of proteins. They're measuring in different buffers. Um, if you try to train a model that doesn't understand all of those labels and understand all of those nuances, the differences between those experiments, you're going to run into a lot of trouble. So really, what a technology like AlphaSeq enables, because of the scale that we're able to measure protein interactions, we can essentially have a single interoperable framework by which we can assemble, billions of measurements that are all run under the same conditions, that all include the same controls, that essentially, generate a data set that is high quality. So we're really thinking about, the quality of an overall data set rather than the quality of an individual data point.
Is the industry underestimating how much wet lab infrastructure is required to unlock the full potential of AI?
I think that the industry is underestimating the amount of data that's going to be required for truly generalizable models. But I also think that it's getting better. So as I mentioned before, I mean, I think that up until recently, there was an assumption that publicly available wet lab data plus clever ML tricks would be enough for solving a lot of these problems. And I think the consensus really has changed, where there is a broad understanding that no, there is a need for kind of dedicated, very large, high quality, interoperable data sets that are going to be needed to solve these really challenging problems. I think that there still is some debate about exactly how much data is going to be needed. But the industry definitely has come around to recognizing that more data will be needed.
How do you scale wet lab throughput without compromising quality?
Really, it's all about assay design, making sure that there are appropriate controls, making sure that assays are being run in a consistent way. And for us, what that means is, AlphaSeq assays are being done under our roof, under a consistent standard operating procedure with identical controls. Again, very different than publicly available protein interaction repositories, which are being done in hundreds or thousands of different ways under many, many different conditions.
You mentioned exploring in-house therapeutic programs. What ultimately led you to prioritize being an industry enabler instead?
Yeah, I think at the end of the day, the question was, how can we have the greatest impact as a company? And what really differentiates us at Alpha is our ability to generate data at scale. So either we could take all of those data, we could train in-house models, we could apply them to a very small select number of therapeutic programs, and have impact that way, which many companies choose to go that route. But really, once we started to see the pull from pharmas, from biotechs, from AI native companies that are emerging, we recognize that there is just such a need, such a hunger across the industry for the type of data that we're able to generate. And we believe sincerely that we can have a much broader impact on hundreds or thousands or tens of thousands of therapeutic programs by helping the ecosystem broadly rather than just focusing internally. Uh, I think another aspect of this is at some point it's really important for a platform company to decide what you are really good at, and make sure that you're building a company that is aligned with that vision, with that objective. So we have built a team. We have surrounded ourselves with investors and advisors who are all best positioned to maximize our impact as an industry enabler. And we really believe that that's the right path forward for the company.
Do you think platform companies will capture more long-term value than asset-centric biotech companies in the AI era?
I think that there's room for both. I think that in our industry, always there's a tremendous amount of value that is captured by assets, and I think that's why a lot of companies choose to focus on internal pipelines. Um, but I think that essentially the foundational capabilities that are needed to be competitive as these AI approaches get more mature are really going to require either every company individually to make massive, massive investments in proprietary data generation and training their own proprietary models, which is going to be very limiting. It's going to be a major, major barrier for new entrants, and is really just going to be a capability that's limited to those most funded new companies and the big players, big pharmas. Or there are going to be companies like Alpha, who essentially serve to provide data, provide models, provide capabilities broadly across the ecosystem, so that we can continue to have new entrants. And I think that there will be kind of clear opportunities for capturing value. Um, but yeah, I mean, I think again, broadly speaking, uh, as an industry enabler, as a platform company, we believe that we can have a significantly greater impact across the industry and across human health.
And how do you think about monetizing data through licensing, partnerships, subscriptions, or other models?
Yeah, so there are a few different models. So we license our data. Again, I mean, coming back, so we have kind of two different core ways that we work with partners. When there is a partner that is focused on a particular therapeutic program or a few therapeutic programs, we will leverage our data generation capabilities, also our in-house models and other know-how, to either discover or optimize assets, which are then wholly owned, by our customers, and they move those assets forward. So that's one model under which we're working with a number of companies. For some programs, like really challenging dual specific engineering, or molecular glue discovery, these are deals that are kind of structured with milestones and royalties. For most kind of more standard antibody optimization campaigns, these are kind of straight fee for service arrangements. And then for data and model training, yeah, we license data. So that is, both we have generated an enormous amount of data in-house, that is pre-existing data that is non-exclusively licensable. And then we also work with companies, with partners, that will come to us and prospectively design experiments, that we will run, and they will license those data. Um, Even with these models, and even with the scale at which we're able to generate data with AlphaSeq at Alpha, um, we still see some limitations, right? So we're talking about data sets on the order of about 1 million affinities in a single AlphaSeq experiment. Um, Kind of coming back to the question of how much data is going to be needed to really train generalizable models. Depending on what you're trying to do, I mean, we believe that in some cases it's going to be hundreds of millions, if not billions, of affinity measurements that are going to be needed to tackle many of these problems. And so we are continuing to explore kind of new business models, and new creative ways, in which we can kind of get more data in the hands of our partners, at kind of reasonable economics. And so there'll be there'll be more to share about that soon. But this is something that we think a lot about internally at Alpha.
There is a wave of AI protein design companies emerging. How do you differentiate A alpha from the rest?
It comes down to the data that we're able to generate and our focus on proprietary data sets. So, we have the largest proprietary internal data set of protein-protein interactions. We are in the process of building a massive database of structures, so antibody antigen validated structures, both positive and negative for model training. Um, so again, I mean, I think that there is a lot of emphasis on using different architectures and different strategies for training models, and that is fantastic. That is moving the industry forward. But at the end of the day, all of these models are going to hit data bottlenecks, and then need to work with companies like Alpha, who have truly differentiated, and sort of, really novel sources of data. That is our key differentiator.
Are we heading towards a world where a small number of companies control foundational biological data sets similar to big tech and AI?
I think that we are trying to avoid a world where data all gets consolidated within a very small number of players, right? So I think that one of the things that we are hoping to enable through Alpha, again, through creative business models and the such, is we can essentially take the the wet lab burden, right? We have this proprietary assay that has been honed and developed, and we've accumulated these very large databases. We want to, yes, enable big pharmas with those data so that they can apply them to all of their therapeutic programs. But we also want to work with the smaller model makers, the smaller biotechs and pharmas, who do not have the budgets or capabilities to generate anything close to the diversity of data that we are able to generate. So our hope is that by companies like Alpha, who are acting as industry enablers, we don't sort of consolidate capabilities into only a very small number of big players. And I think this really comes down to we believe that innovation in biopharma has historically happened both within big pharmas, but also to a very large degree in academia, in smaller biotechs. Um, if the kind of requirement, if the kind of starting point uh, for having an impact in our industry is that you first have to have hundreds of millions of dollars in investment to train a cutting edge model, um, it's really going to chop the bottom off of that innovation ecosystem. And I think that would be a disservice to us all.
You've worked with organizations like Lawrence Livermore National Laboratory, the Department of Defense, the National Science Foundation, and the Gates Foundation. How have these collaborations shaped your platform?
So in all cases, working with the government, working with nonprofits, these have been an opportunity for us to develop and kind of better understand our platform and our data. These have been great opportunities for us to generate data that is essentially co-owned, right? That's used by our government or nonprofit partners, but also data that we can use internally for improving our own capabilities and training our own models. Um, I think for many of these collaborations, we've been able to focus on infectious disease applications, where measuring protein interactions in a library on library scale is key, right? So you need to be able to look at how a diversity of therapeutics bind to a diversity of viral variants, for example. And so it's a great kind of test bed for generating data that is valuable for solving real world problems, but not necessarily the ones that are kind of the largest economic opportunities. But still, the data that we generate through all of that work is equally valuable for training models that can be applied to other therapeutic areas and therefore can kind of improve the way that we can work with other partners and uh, again, yeah, uh, apply to other indications and other applications.
So, do those types of collaborations push you toward different use cases than commercial partnerships?
In some cases, yes. So I think the infectious disease kind of space is a great example, right? Where with the Gates Foundation, with Lawrence Livermore National Lab, and the DoD, largely those collaborations have focused on either kind of ongoing pandemics or potential future biothreats or other infectious disease related applications. Um, there have been some other, kind of partnerships. So our work with Gilead, for example, in HIV was also kind of very much in that space, but there are less commercial opportunities in the infectious disease space. But all of those data, right, where we're looking at diversity of binders and diversity of targets, are equally valuable for all of the other model training efforts that we have ongoing at Alpha. So it turns out that by sort of being non-competitive economically, but being just as valuable from a model training perspective, these have been very powerful synergistic relationships.
What would you say the biggest misconceptions investors and operators still have about AI and drug discovery?
Probably the biggest misconception is that AI is going to replace or displace experimentation. We will always need wet lab. We will always need ground truth. And if anything, probably as models improve, it only makes companies, makes the industry hungrier for wet lab validation data to really inform whether models are performing, and where they might be not performing. How can we generate the right data sets to get them back on track? So I think that the way that we see this is very much that there is going to be a long term synergy between computation and wet lab. And people should not expect that as AIs improve, that wet lab needs are going to go down. I think we're yeah we've certainly seen this in other industries as well, right? Where as the computation improves, it doesn't kind of displace the need for data. It actually increases the need for data.
And how has your view of the intersection between biology and machine learning evolved since founding A Alpha?
Yeah, so I think at the very beginning when A Alpha was kind of first conceived, there were kind of hypotheses around how we could use our data plus kind of next generation computational tools, but it was really kind of the early days for applying ML. We were still extracting a lot of value from our data in a quite manual way. So, for example, right, I mean, we can do multiplex epitope mapping, right, where we take thousands of antibodies and we build a mutational library of an antigen and we cross those two libraries. And then, without any machine learning, we can determine the epitopes for all of those 1,000+ antibodies. That's a useful application of the platform that doesn't require particularly sophisticated computation. And that's largely where we started, right? I mean, we knew that the data that we were generating would allow us to gain these types of insights at a scale that otherwise wouldn't be possible. I think where we've seen ML evolve, it's now 100% intertwined with everything that we do. Right, we are able to extract so much value, so much more value out of the data that we generate, and then of course we're also designing experiments with the ML objectives in mind, in a way that we were not able to do when we first started the company. So now we're moving from kind of getting discrete value out of each experiment to what are the next 10, 20, 100 experiments that we should be running, that are going to be maximally impactful for training a generalizable de novo antibody design model, for example, or a generalizable zero-shot antibody optimization model. It's just sort of a different scale of thinking about some of these problems, which is, in my mind, just very, very exciting.
So, as you look out over the next several years, what will AI driven protein engineering enable that maybe today feels unrealistic?
I think that AI-enabled protein engineering is going to really allow the field to tackle problems that were completely out of reach just a couple of years ago. I think the first place that people's mind often goes when you think about, kind of, we used to do things experimentally and now we're moving to doing them computationally, is that it's going to save time. And to some extent that might be true, right? There are some workflows that are going to go from multiple experimental iterations down to a day or two of compute. But really, in the overall timeline of getting a drug to clinic, those time savings are relatively speaking quite minimal. I think the place where I am really excited, where I think the industry is kind of really kind of poised for impact, is again, yeah, the ability to engineer drugs that just otherwise would not have been possible. So, here we're talking about engineering binders to bind to epitopes or bind to targets that you could run thousands of phage campaigns, you could do thousands of immunizations, and you would not find an antibody that binds to this particular epitope in this particular way that we need for a therapeutic. So, an old school kind of guess and check approach just never would have worked. But now that we can screen, billions of designs in silico, we can actually find a binder to that epitope, and unlock the ability to engineer that particular therapeutic. We can engineer a biparatopic where we have a single arm of an antibody, that in a single paratope can simultaneously bind to two different antigens, in a way that again, trying to find something like that through conventional phage panning or immunization or other kind of traditional antibody discovery approaches would have just been so unlikely that it probably may not have been worth doing. But because we have the ability to engineer these things intentionally in silico, we can now again, and engineer these new drugs that otherwise just would not have been possible.
David Younger, co-founder and CEO of Alpha Bio. David, thanks so much for your time today.
Thank you so much. Enjoyed the conversation.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.